I'm Benjamin Epstein. I'm a graduate student here in the laboratory, and I work on gene therapy for genetic diseases of the retina, so diseases that make people blind. I kind of fell in love with biology in high school. When I first took biology, I was kind of bored by all of the details and facts. But then I took an anatomy and physiology class as a senior and had to do a project on the immune system. And when I started researching and trying to explain it all, I realized that I had to draw upon all of those details and facts that I knew. And I then had a context for why they were important. And all of a sudden they seemed really cool because all of those technical facts about proteins and hydrogen bonds and whatever became relevant to how your body fights off disease. There's a personal connection as well. I spent a decent amount of time in the hospital in middle school. You know, there was a little bit of a sense that people shouldn't have to be going through this, and we should be able to do something about it. And to the extent that the doctors could do something about it, I was very grateful to that. And so there's an extent to which I was excited at the possibility of being able to work towards ensuring that no one else had to go through what I went through.
But not go to medical school and become a doctor.
Yeah, the key to the problem was not that I didn't have skilled doctors, but that we didn't know what to do about what I had. It was a lack of knowledge, of understanding of what was going on. So what that motivated me towards was the field of research where I could work towards understanding more and giving the doctors, essentially giving doctors more tools to work with to treat people. I'm working in gene therapy for treating genetic diseases, and the way we do that is by making viruses that carry DNA that is not normal virus DNA, but it's replacement DNA for people who have a genetic defect. Once I've assembled a piece of DNA, then I go to the experimental phase with cells in a dish. We have lines of human cells to practice on, and usually I'll then go and take this DNA and try putting it directly without any viruses into some cells and seeing if I can get that DNA to function, to do what it's supposed to do. Once I've verified that it works, it does what it's supposed to do, then I package that DNA into a viral shell, and these cells assemble a virus. I wait a couple of days and collect all of the virus that those cells have produced for me, and then I try to apply it to some sick cells and see if it can do what it's supposed to do.
That's amazing.
It's very exciting. In the short term, sometimes it can be frustrating. You can work day after day and get no results or have an experiment inexplicably fail, and it can be very tedious and frustrating and demotivating. But also in the short term, day in, day out of failed experiments, one successful experiment, one tricky technique that you succeed in making work, that you connect two pieces of DNA that you were having trouble connecting together, that you manage to extract a protein you were having so much trouble extracting, even if it's just step one in a 20-step experiment, that little bit of success can have me jumping up and down for joy.
It's like, I don't think that was there before.
Science is very collaborative, and I work in a lab with a lot of other people, and I'm surrounded by people who are all working on different projects. I spent like a month and a half trying to clone this one piece into this other piece. Yeah. When I'm struggling, when I'm having trouble with something, I just turn around, say, "Hey, David, I'm having trouble with with making this virus over here. Can I talk to you for 2 minutes and see if you can give me any tips?"
You might want to try the Golden Gate reaction.
And usually David has a suggestion for me. You're piecing out this the amount of creativity you can get from introducing other brains into the mix just transforms the scope of what you're able to do and increases the breadth of what you're able to accomplish. It also makes the enterprise a lot less lonely and a lot more enjoyable, having other people around to work with, to help with, to commiserate with, to be excited when you get an exciting result. It makes for a good environment. I've been working on a disease called retinitis pigmentosa, which is a genetic blinding disease. About 3 weeks ago, I got an email from a friend of mine from college that said, "Dear friends and family, as some of you might have heard, I am going blind from a disease called retinitis pigmentosa." And I sent her an email back and said, "I know exactly what you're talking about." And she was working to raise money for research. And I said, "I'm happy to give you some money. And I'm also happy to tell you that my work day in and day out is literally to find a cure for you." I think I am doing work that can help the world. And that's why I do it. Because I think I can help people like my friend Erica, like myself as a little kid. I think there's a lot of work to be done to change the way we do things now and invent and understand things in ways that we haven't before. When I come in and do this work and study these cells and look at these viruses, I am amazed at the diversity of creation out there and of the wonder of this world and of life. We pray for God to heal people who are sick. You know, whenever I imagine that prayer goes up to heaven and God hears that prayer, that please help my daughter who is going blind, the way I view it, God says, "All right, I've got Benjamin on that duty. Here you go."